Why don't we get started? Thank you all for coming to the Brain Body Lecture Series. To keep in the spirit of such an amazing session, we've got Brian Kim here to visit and to give his lecture to us today on the sensation and perception of itch. So, I don't know why we have these balloons here, but I think it's somebody's birthday party today, so we're here to celebrate. So anyways, we recruited Brian Kim here about a year ago from WashU. He's come in to develop and lead an amazing program on sensation, itch, and immunology. His work spans the gamut of neuroscience and immunology and dermatology. So I think in the spirit of brain and body, Brian really encapsulates that really complex interface between neural circuits and peripheral systems and immunology. So he's been doing really groundbreaking research on understanding how sensory nerves can co-opt kind of classic neurotransmitter signaling pathways, the things that we all neuroscientists study on a regular basis, to control local neuroinflammatory processes. It really is kind of an outside of the box, innovative, new way of thinking about how sensory nerves or neuroinflammation in general can operate. So without further ado, I'd love to bring up to the stage Dr. Brian Kim to present his work. Thanks again for coming.
Yeah, thank you. Thanks to Scott Russo. It's great to see people in the flesh as well. Yeah, congrats to Scott on the formation of this center. I was very excited to hear about the center because I think it's very timely. And I think a lot of the stuff that we do is inadvertently really kind of integrating into this. And I'll try to speak to that a little bit throughout the talk. And yeah, we'll just get right into it. I have a number of disclosures, quite a number, as itch has really become a big topic in the last 5 years in particular. So the first kind of fundamental question is, for us, has become, how do we sense the world around us? And how does this inform itch and perhaps even other sensations? And that's how I'll try to kind of end the talk. But as an immunologist, it's interesting because most of what I'm talking about really falls within the domain of neuroscience traditionally. And as we know, when we think of the five senses, there have been a number of Nobel Prizes awarded in these different domains. And most recently, the Nobel Prize in Physiology and Medicine this fall to David Julius and Artem Patapoutian for the discovery of how we sense temperature via trip channels and touch via the piezo receptors. And it's really obvious immediately as to why the five senses are important for the most part. But it's funny, when we think of the five senses, we don't even really immediately think of pain and certainly not itch, which probably falls within the domain of touch. But what happens when we actually have mutations in receptors for pain? It's actually, might think at first that this is actually a bit of a superpower, but it really isn't. We know that Ashley Blocker, shown here, who actually was born with a mutation and can't really sense pain very well, would injure herself. And in fact, as a toddler, would bite her fingers. And that is why she's actually bandaged in this picture. So actually, pain, of course, is very, very important as a protective measure so that we know how to avoid certain kinds of traumatic or harmful stimuli. But then it becomes even less clear, though, as to why we itch. And this is something I'm probably asked the most when I give a talk, is why we actually itch. The short answer is we don't exactly know, but we can actually speculate. There's probably two, at least minimal reasons. One is to actually sense certain kinds of noxious stimuli, such as disease-borne insects in the environment, in a way that's not so aversive, like a bee sting. So that's not immediately makes you want to avoid the environment, but allows you to sense the environment, feel that something's wrong, and still remain within perhaps a limited amount of time. But the other that we appreciate more and more in modern day is that as we get older, as organs fail, as different parts of the body fail, actually itch is actually a final common pathway. And it's why it's such a common symptom as a chronic pathology. And actually itch is also in a way to tell ourselves that something might actually be wrong. So in a way, itch has an exteroceptive component to sense things from the outside world, but also from inside the body that there's something wrong as well. So there's an interoceptive component to it as well. So we think, but this is speculation. But one thing's for sure is that itch is highly conserved, and it's really thought that all vertebrates itch. So this evolution has not done away with the sensation. But notwithstanding the kind of homeostatic and reasons for why we need certain things like sensation, but what I spend most of my time as a physician doing is understanding what happens when these things go really awry. And I actually, believe it or not, spent most of my career studying things that were seemingly vestigial, like innate lymphoid cells and basophils. And the one criticism sometimes is that you might be studying something vestigial, but the issue is that even in modern days, things that are seemingly vestigial can cause a tremendous amount of problem for patients if these things actually are turned on and can't turn off. And here is just a small list of conditions in which itch is actually central morbidity. These include atopic dermatitis or eczema, which I'll speak to. Other conditions like notalgia paresthetica, where you itch in the middle of the back. It's why the back scratcher was invented. Prurigo nodularis. They range from inflammatory to more neuropathic. There's also uremic pruritus. Patients who are on dialysis will itch with high penetrance. I'm not going to go over all the different conditions here, but really just to list that there are many, many itch conditions, and some of them will come back later in the talk. So when we think about itch, how does the skin sense itch? It's actually interesting because again, I'm not a neuroscientist, so I actually really was, when we think about the peripheral nervous system, I was originally much more acquainted with the autonomic nervous system. And I think that was because I was trained as a physician. So the idea was that heart rate, breathing rate, gastrointestinal motility, these are all things that in an efferent fashion regulate the different physiologies within tissues. But ironically, despite the fact that on the right, the somatosensory nervous system is essentially mostly evolved for the skin, I didn't really think about this a lot. And it's not something in the field of dermatology, for example, we are taught or focused on. But in very, very overly simplistic terms, almost embarrassingly simplistic, and from a neuroscience standpoint, we know that the somatosensory nervous system has different kinds of nerve fibers that can sense things from mechanical sensation by way of A beta fibers to A delta fibers. And as we get to the polymodal unmyelinated C fibers, we find that they are truly quite polymodal and can sense everything from pain, itch, and temperature, and a whole host of different sensory modalities. And it's really just a subset of these C fibers, a small subset that actually mediate itch. And the question, though, is why has not itch been studied for very long? Why is this such a new area? In many ways, itch was actually a very kind of trivialized sensation. It's not one that physicians in particular took very seriously. But what's changed and why we are taking it much more seriously, it really starts with the science. And it's really the discovery of fairly itch selective, if not even itch specific pathways, such as the mast-related G protein coupled receptors in the periphery, a whole family of G protein coupled receptors that mediate itch, and perhaps other processes as well. And then also the discovery by Mark Hoon's group of brain natriuretic peptide as being a major neuropeptide from these peripheral neurons dumping into the spinal cord and being very itch specific. This discovery of gastrin-releasing peptide receptor, as well as those neurons being very itch specific in the spinal cord circuit. And then additionally, people are now working on identifying foci within the brain that actually receive these itch signals for perception. But it's really with the scientific accomplishments that it then made us believe that itch was actually tractable. A misconception was that itch was simply a mild form of pain. So it wasn't going to be a specific, distinct molecular target, but all of this was overturned with these discoveries. And I think it really put itch biology on the forefront. And what I'm going to talk about later is how we've had a lot of actually therapeutic success in a very short amount of time. So our laboratory has always been focused on understanding what are the initial events that trigger inflammation at a barrier surface, and using skin as a model paradigm. And that was really where most of my postdoctoral work focused on. But when I started my laboratory, we became really interested in understanding how does inflammation trigger something like itch, and how can this represent a model in which inflammation and neurodysfunction, or perhaps even us sensing inflammation, can occur. And this can be kind of a way to understand this through itch. And to study this, we used atopic dermatitis, or eczema, as a model disease. It's one that I started treating very early on in my career in a specialized manner. It's very common. If you don't have it, you know someone who has it, and very likely a family member. In its severe forms, can be incredibly debilitating. It's an atopic disorder, so it actually clusters with other allergic disorders like food allergy, asthma, and other emerging conditions as well, such as eosinophilic esophagitis. But when we first started studying this disease, it was actually very limited in what we could do. Topical steroids are kind of the mainstay therapy. It was not one that dermatologists necessarily enjoyed treating because it was difficult, but that's changed quite a bit in the last few years. But itch was always the central symptom, and it is certainly the major morbidity. So when we first came to study this disease using more of an immunological lens, we knew a few things. We knew that there are allergenic or environmental stimuli that can activate this arm of the immune system, the Th2 cell axis. These are T cells that have antigen-specific receptors, but still what the antigen is remains very elusive. But what we knew was that in a modular fashion, what we referred to back then as Th2 cell-associated cytokines, IL-4, 5, and 13, will actually trigger a whole cascade of inflammatory events, including the production of IgE. IgE is reacted to a variety of different environmental allergens in all the atopic disorders. And then also these cytokines can directly damage the barrier. But we became interested in actually understanding how the barrier itself can directly engage with the immune system. And the clues came by way of these cytokines, alarmin cytokines like IL-33 and TSLP, which actually immediately can sense a barrier breach and then is released from the barrier. And we knew at that time that these cytokines were potently activating these pathways. And this led to the kind of my early work, which is understanding then that innate cells like group 2 innate lymphoid cells, or ILC2s, which are hardwired during embryonic development into different tissues, we discovered them for the first time in the skin and as being major sources of IL-5 and 13, but also that rare circulating basophils, which when I was in medical school, I was taught it was a vestigial cell, as a vestigial kind of relative of the mast cell, actually come into skin and are major sources of IL-4. And this, in recent years, after I started my laboratory, we came to appreciate that these canonical, what we now refer to as type 2 cytokines, because innate cells actually are major sources of them, not just the adaptive Th2 cell, that these canonical type 2 cytokines can actually directly communicate with the sensory nervous system. They employ downstream JAK signaling pathways that we typically think about in immunology more than neuroscience to actually mediate these kinds of processes. And now we know that the therapeutic blockade of these cytokines is very, very important across in a pan-allergic fashion. But just to highlight some of the ways in which you can kind of approach this this is actually my first graduate student, Landon Etchen. We took very kind of low-tech approach. We just simply extracted dorsal root ganglia, and in collaboration with Steve Davidson, we're able to get cadaveric human donor DRG and just look at by RT-PCR for whether they express these different receptor components. In fact, they do. IL-4 receptor alpha, which is the receptor for IL-4 and 13. We can also detect IL-13 receptor alpha-1, and then the canonical kind of itch or pruritogen receptor, IL-31 receptor, we could also just use as a positive control to identify within the sensory nervous system. We can never detect IL-5 receptor in the DRG, though it's been reported within the actual nodose ganglia that it is expressed, indicating that maybe actually some functional heterogeneity across different tissues. But we just did kind of traditional calcium imaging to then test whether these cytokines are actually functional on these sensory neurons, and we can then detect a response to recombinant IL-4 in mouse DRG as well as human DRG. And you can see here that this neuron is not only responsive to IL-4, but also capsaicin, indicating that, in fact, this is a TRPV1 positive neuron. And we modeled this in mice, and we used a very now well-established model of atopic dermatitis-like disease in which mice are treated with a vitamin D analog. It actually induces expression of these alarmin cytokines, and then it potently activates the immunological events that are mirrored in human atopic dermatitis. They develop rash, histopathologic features, and immunologic cellular features. But if we just aren't just bulk RNA sequencing in the skin, with the induction of this disease, we see key type 2 cytokines, the alarmin cytokines, chemokines, and keratin alterations that have been already well reported in human atopic dermatitis, particularly from translational groups like Emma Gutman here at Mount Sinai. But importantly for us, these mice develop behavioral itch, and they develop scratching behavior that's highly elevated and chronically so. So whenever you look at these mice, they are spontaneously scratching at any moment. The great thing about studying itch in mice is that it's very objective, because they have no idea that that's what you're actually doing. In humans, the problem is a lot of kind of cortical processing can affect itch quite a bit, but in mice, you can eliminate that element. And so the way we kind of interrogate this is we use sodium channel 1.8 Cre mice, and then we can flux out different receptors in the peripheral sensory neuronal compartment. And in this case, we're able to actually delete IL-4 receptor alpha just in the peripheral sensory neurons. And we found that, in fact, that the scratching bouts are greatly reduced, but also the disease severity. And I'm actually going to come back to this point. So it's been a number of years since we did this work. Interestingly, a question that was brought up by the reviewer was, is this really due to a reduction in scratching, or is this really is this something independent of the scratching pathology? And it was actually a really good point. We didn't really want to get into that at that point, but I'm going to come back to this in a second. But we know that drugs, monoclonal antibodies that actually target this receptor are highly effective. They actually have unprecedented efficacy for atopic dermatitis. And actually, they have really revealed how important itch is in these diseases and have had unprecedented effects on the attenuation of itch. In the interest of time and showing this, the way we think that actually these particularly IL-4 and 13 work is that they actually sensitize the nerve to a whole host of other pruritogens. So it's not so much that these cytokines are all pruritogens and necessarily slam the nerve and cause a lot of itch, but they actually, what we think is they actually change the membrane depolarization in a way, they depolarize the membrane in a way that make them much more sensitive to a whole host of stimuli. So the simple way to think about what this drug might do is may actually anesthetize the nerve to a whole host of other different stimuli. But in parallel to the work we're doing, which we're taking a very kind of candidate-based approach, that other groups like Patrick Ernfor's neuroscience groups were actually just doing single-cell RNA sequencing across different ganglia, and I'll show this here in a bit how this kind of plays out. But I'd like to think of them as somewhat dispassionate towards itch, that they're very objective neuroscientists, and they were finding that some of the most readily identifiable neurons at the single-cell level are actually these itch-specific neurons or pruriceptors, and they would classify them as and I won't get into this, but non-peptidergic 1, NP2, NP3, and I just color-coded them for you. And in fact, what was great is that the single-cell studies actually confirmed a lot of traditional functional studies, suggesting already that there's modularity of these neurons with regard to how they respond to different pruritogens. So it's not just every nerve responds to the same thing, but one of the things that Landon observed early on was that the IL-4 receptor alpha was selectively expressed on these itch neurons, but also very broadly. So across these different subsets. And that actually provoked a clinical hypothesis for us that putting this all together, we thought maybe it's not just an atopic dermatitis story. Maybe that the fact that these type 2 cytokines are so conserved, that maybe this is more of an itch phenomenon, and we can actually broadly target many different kinds of itch disorders. And one condition we're interested in is a condition called prurigo nodularis. This is actually very different from atopic dermatitis, where you start with inflammation or a rash in the skin that leads to itch. These patients actually are primarily itchy, and then they secondarily damage their barrier to give themselves very distinct nodules. This is a patient of mine. This is actually not very dramatic. If you Google it, it's incredibly dramatic what they can do themselves. The idea here is if you actually duct tape these patients, they would never develop these nodules. They'd be very, very distressed, but they wouldn't develop these nodules. So it's really this kind of scratching reflex that actually triggers a lot of pathology. Of course, there's inflammation once they trigger this kind of pathology, but the primary genesis of this is thought to be itch. And so what we actually proposed to Regeneron, who actually had pioneered the development of dupilumab early on, was we said, we think that this drug should be used towards patients with other itch conditions like prurigo nodularis. At the time, the company was not really aware of what this was. This is the itch landscape. We're really at the leading edge of even defining these conditions. And what happened was actually I designed an investigator-initiated trial. I'm not much of a clinical trialist, but I felt that we should really push this. Went back and forth. They agreed to fund it. But what happened at that point was there were enough case reports coming out that dupilumab was effective for prurigo nodularis that they decided to go straight to phase 3. And in fact, the phase 3 was just, was a home run, met all the key primary and secondary endpoints. This was reported in the fall. The prediction is this drug will be approved by the end of the year. So this is fantastic because you have a condition that was ill-defined, no FDA-approved conditions. And we're really, if it weren't for the science, we would not have been able to kind of embolden this kind of exploration, even in the clinical space. So I think this is a good example that the science really, the thread here is that the science is what's really leading the way and at every step. But also we see patients, what I see mostly now in my clinic on the Upper East Side is chronic pruritus of unknown origin. These are actually patients who are more elderly. The same way that a lot of elderly individuals will start to develop a lot of neurodysfunction, they start to itch. They don't actually have much in the way of an atopic history. They're incredibly debilitated by this because there's no adaptation. Atopic dermatitis patients from childhood have itch, so they actually have a lot of plasticity. They can actually adapt to it. These patients are very, very distressed, often suicidal. And they look like this. So they just scratch where they can reach, and then where they can't, they spare themselves. And then anecdotally, out of our clinic, off-label patients who were treated with dupilumab actually improved. We did a retrospective study to look at over a 30-month period just out of my clinic, all the patients who had severe itch, 7 or greater over time, and they all did quite well. So as you can imagine, being a bit of an instigator, we were pushing really hard for Regeneron to do this as well. And so they've partnered with Sanofi and for this drug, and now they've actually opened up another clinical trial which started enrolling in March. So I hope that in a year or so, I'll report back a very similar kind of positive outcome. But at the time, independent of this IL-4 story, one of the things that we did know is that dupilumab was not approved. There's no blocker for this receptor, but there were JAK inhibitors, 2 JAK inhibitors, tofacitinib and ruxolitinib, one for rheumatoid arthritis and ruxolitinib for myeloproliferative disorders. And if you look at the single-cell RNA sequencing data sets, you find that actually these itch nerves actually do highly express downstream JAK1. And we know that many of these cytokine receptors are in fact dependent on JAK1 downstream. But you zoom to just in the last year, and other groups like Steve Davidson here and Ted Price have actually identified that there are itch-specific neurons also within human DRG as well that are readily identified. And one of the things that we can't quite figure out is why they keep coming up so cleanly in these single-cell sets. It's kind of interesting. They're actually, the thing we're a little perplexed by is why they're so well represented. And I have some theories about that. So, but one of the things you see JAK1 here comes up very readily. So we think that there's a lot that's translating from the mouse to human. I think we're very lucky. And a lot of this is due to the fact that type 2 immunity is a very ancient arm of the immune system, and itch is a very primitive kind of reflex. But we took a similar approach here, and we deleted JAK1 in the peripheral neurons. And here's where it differs. We see no effect on the inflammation. We see exquisite effect on the itch, but no effect on the inflammation, in contrast to when we deleted alpha receptor alpha. So that reviewer was right. Had they really pressed it, we would have been in a lot of, it would have caused a lot of angst back then. But you know, we kind of come back to this concept, and I'll address what I think is going on. And what we actually know is that many cytokines go through JAK1. And the idea was really kind of medieval. We thought, okay, we've identified, it took us several years to publish a paper on 1 or 2 cytokines being involved in itch in a very specific way. If we add up every cytokine, this is going to take forever. We want to just try to cure itch or treat itch, right? So we wanted to go downstream to just really choke it off. And one of the predictions we made, we like to make a lot of predictions based on the mouse modeling, is that we thought that because of the breadth, there would be broad activity, rapid, because you're choking off more, and potency with this. And then you zoom to the last year, and the phase 3 clinical trials have gone actually head to head. So the JAK1 selective inhibitor, or upadacitinib, went head to head with dupilumab. It actually, this downslope here shows that, in fact, much more rapid reduction in itch. And then abrocitinib is another JAK1 selective inhibitor. Now, with the upstroke here, you see that high dose of abrocitinib does outpace dupilumab, shown here in green. It does catch up, though, later on. So there's probably other things going on as well. But we actually early on shared a lot of our mouse data with the JAK effects on the nerve with Insight Corporation, and actually we were trying to get actually oral ruxolitinib for our patients with this chronic pruritus. And in these conversations, what actually ended up happening was a bit of a bait and switch. We ended up designing a phase 2 dose ranging study with topical ruxolitinib for atopic dermatitis. But the nice thing was we could actually design the trial in a way that we were reading out itch in a very high resolution manner, because we knew what we were trying to look for here. And not just as an anti-inflammatory agent. And this drug is now approved as of last fall. And it was actually the first JAK inhibitor approved in the field of dermatology. So this was another kind of big win that kind of fell out of the science. But we were able to get tofacitinib for these patients with CPUO. And again, just scratch marks where they're itching. The scratch marks go away. Very anecdotal cohorts of severe patients before and after. This is by no means anywhere near our clinical trial. But of course, as you would imagine, companies are now, JAK inhibitor companies are now approaching us about whether they should go forward with this in light of the fact that Regeneron keeps kind of moving ahead very quickly. But I think what we're appreciating is that there's, if you think about itch, we think of it in a very simplistic terms, that these cytokines hit the receptors and they can trigger itch or amplify itch. And it's just this kind of unidirectional kind of afferent mechanism. But what we also know is that these itch nerves actually are very much peptidergic. So they actually have the capacity to release neuropeptides into the tissue, like CGRP, but other neuropeptides as well. And depending on the receptor and depending on the way in which this nerve is hit, we think is that there are ways in which neuropeptides are released to either activate or even, for that matter, negatively regulate inflammation. And so really understanding at the receptor level what's going on actually is very important. And that's why we think actually the JAK1 deletion is, there's a discrepancy with the IL-4 receptor alpha. And we are understanding this. We have data to support this concept now. And it's a very interesting area. And the other thing to consider here is that there are many different sensory opathies, right? There's pain, there's itch, there's irritation across the tissue, across the body. And whether this kind of, we always think of this irritation as a subjective phenomenon where we're often clinically a bit dismissive of. We want things that are really objective: histology, biopsy, radiology. But the question we have to ask is, whether it's rheumatology or these other fields, whether a chronic kind of pathologic sensory state itself, in and of itself, could that be pro-inflammatory? And this is something I think we have to really consider. And actually, along the lines of the center, does this work its way all the way up to the brain, right? So, can some focus in the brain thread all the way back to the skin and promote other kinds of inflammatory disorders? So I think this is a concept I think that's kind of on the frontier. But what we're saying is that cytokine blockade, JAK inhibition, we know that this is anti-inflammatory, but what we're really proposing very strongly now is that these are just as much neuromodulators, and they can actually be tailored to be even more neuromodulatory if that's how we think of these agents. And this is certainly the way the companies are thinking of it. In fact, they're even marketing increasingly the drugs in this fashion as well. Again, they weren't conceived that way at all. In the interest of time, not showing this work, but I just wanted to highlight, what I've talked about so far is in the setting of chronic inflammation, how local cytokine processes will drive through JAK or other pathways, chronic itch at this neuroimmune interface. But what we've also identified in diseases like atopic dermatitis is that the systemic inflammation will actually trigger systemic activation of basophils. And this will actually result in the basophils being activated and primed in a way that they respond to IgE and override the response of mast cells in the tissue and actually activate a totally alternative leukotriene C4 circuit. So this has nothing to do with kind of classical histamine as the itch mediator. And it starts to explain heterogeneity of even IgE responses, so kind of classic allergic pathways. And this seems to be a very, very potent kind of peritogenic pathway. And I'm not the only one saying this. This was discovered by Mark Hoon, who showed using chemogenic activation of mast cells that this pathway is very important. And recently, Isaac Chewett as well from Harvard Medical School. So we don't generally believe our data until other people reproduce it. and so I'm starting to get very bullish on this pathway now in light of the work of others. But also, one of the things that I want to highlight, I mentioned the MRGPRs that actually led to discovery of and the conceptual advance that there are itch-specific receptors in the peripheral sensory nervous system, but some of them do land on the immune system as well. And what's interesting, what I think is quite beautiful actually, is that MRGPR-B2 in mice is very specific for mast cells. And mast cells have always been associated with neurons. And actually, my buddy Brad Undum, one of my favorite neuroscientists, says he considers mast cells a neuron. I kind of agree with him. And what's interesting is that MRGPR-B2 is on a host of different mast cells throughout the body, and it's very selective. MRGPR-X2 is the ortholog in humans. So there are actually less MRGPRs in humans, which also indicates, we think, that there's much more polymodality of these receptors. What's interesting about MRGPR-B2 is that it explains a lot of reactivity that we misattributed to IgE, to a whole host of different kinds of allergens. Turns out many different endogenous, exogenous peptides, as well as drugs, synthetic drugs, can activate this pathway on mast cells and can cause non-histaminergic itch, also kind of pseudo-anaphylactic reactions. So it's actually explaining a whole new paradigm of allergy. But there's also another receptor, MRGPR-A6 in mice, that's only on basophils. And this is something that we're exploring and as well. And what's interesting is if you take MRGPR-A6 reporter mice, this is in collaboration with Xinzong Dong, that if we gate on the MRGPR-A6 cells, actually they're entirely all basophils. They're not mast cells or eosinophils. So they're c-KIT negative, they're c-KIT negative. But also, as I mentioned, the kind of contracted repertoire of MRGPRs in humans, what we think is actually MRGPRX2, if we actually gate on human blood, there's actually, these are also Fc epsilon receptor CD123 positive basophils, largely. So what we think is that MRGPR-B2 and A6 being divergent and specific for mast cells and basophils in mice are actually shared as MRGPRX2 in humans, and represent the receptor, at least in part, on basophils in circulation and mast cells in tissues. And this is an interesting concept. We're struggling to get an R1 funded on this. People have very strong opinions about this, so it just gets clobbered, but eventually I'm sure we'll get it through. But anyway, that's something we're exploring. But one of the things that I want to highlight is that the cis-LTR2, does it pan out also within in human DRG? And if you go back to Steve Davidson's data sets, you can see these are the itch neurons, IL-31, RA positive, the co-receptor OSMR. But yeah, you find cis-LTR2 is very much, in fact, enriched on these neurons, as well as the brain natriuretic peptide that Mark Hoon discovered. So this is just, again, just trying to cross-validate. We really love to mine other people's data sets. But I want to just pivot in the last part is to moving beyond kind of classical pruritogens and talking about some of the opioid signaling pathways as well. And one of the things that we at least appreciate within the skin, and it's been well established by people like Sarah Ross and others, was that that there's a balance between the mu opioid and the kappa opioid pathway. So these are very distinct pathways. And the mu opioid pathway, we do know even clinically, often triggers itch. So if people get morphine, whether in the periphery or even in the spinal cord, it can actually trigger itch very intensely. And we can talk about why that is. In part, it's because the actual, probably the C fibers do respond to this, to the mu opioids. But the kappa opioid is actually counterbalancing. So actually agonizing the kappa opioid receptor pathway actually tends to counterbalance the mu opioid pathway, and has been considered very much an itch target for a long time. And it's really to kind of balance this out within the skin. But what's interesting is that, and there are actually drugs that aren't necessarily the most optimal, like naltrexone, that block the mu opioid, and it's been used as not a great strategy, but a bit of a desperate strategy to treat itch. And then there are other drugs that have been around for a while, like butorphanol, which activate the kappa opioid, but also antagonize the mu opioid. I actually use this in the clinic. It works here and there. There's a new drug called nalbuphine, which I'll come back to in a bit, which is similar, but much better, with much better kind of pharmacokinetics. And then there's difelikefalin. Now, furafine, I think, is approved actually in Japan for uremic itch in patients on dialysis. So there are agents here, and I'm going to speak to this a little bit. But if you actually go back to the single-cell RNA sequencing data sets, what's interesting is that if you look at the kappa opioid receptor, we actually don't find, although it's been reported, we don't actually find it. We can't detect it within the C fibers. They tend to be mainly on the mechanoreceptors, whereas in contrast, the mu opioid receptor is more on these C fibers. So they're kind of inverted. This could be a bit controversial because, as I mentioned, others have reported that it's on these C fibers and it might actually be acting directly on them. But again, this is not our data, but this kind of suggested to us there might be a slightly different mechanism of action, and this is something we're exploring. But notwithstanding that, the difelikefalin, the pure kappa opioid receptor agonist, has actually been approved in the last year for moderate to severe pruritus in patients on dialysis. And but we also were working with Clara Therapeutics and exploring this in atopic dermatitis. What we really thought was this was primarily just a neuromodulator, but this was something that we were trying to kind of settle. And if these mice actually develop AD-like disease, what we find is that if we treat them with high-dose difelikefalin, no effect on the inflammation. If we do flow cytometry, we see no effect as well. But one of the things we noticed is that DFK was incredibly potent. Actually, we see a difference at baseline even in terms of scratch. It was shockingly potent to me. I was really surprised, to be honest. And so then this is a work from Masato Tamari in the lab. And so we just looked within 30 minutes, we see actually a reduction in the itch. So we think, okay, there's no way this is anti-inflammatory. This is just way too fast. And again, we saw no effect on the inflammation. And in parallel, while we were doing this, Clara was running a phase 2 dose ranging study in atopic dermatitis. And one of the things I said to them is, I don't think a neuromodulator in atopic dermatitis is a great idea, because you have inflammation hitting the nerve, lots of pruritogens, seems like an uphill battle. The reason why the other drugs I mentioned are working so well is they are still anti-inflammatory. Sure, they're neuromodulatory, but they're still taking care of the root cause. With a pure neuromodulatory drug in an inflammatory disorder, this is probably not the greatest idea. So what they baked into the trial was another primary endpoint, is to look at patients with milder inflammation, so less than 10% body surface area. Well, it turned out that was the only endpoint in which it actually works. So you actually have to get to the milder disease to see an effect of the drug. So again, this is all informed by our mouse work. So so I get very upset when people say mouse study models don't matter because we've been doing this a long time now, and helping companies do the right thing, because these trials are incredibly expensive. And but in parallel, they also were very responsive to me. I said, you guys should do a purely neuropathic itch condition like notalgia paresthetica, which is not inflammatory. You itch on the back. It's why the back scratcher was invented. There's probably people in this room that have it. They didn't know it until I mentioned it. And some people, it's very severe. They can't concentrate. They have trouble focusing. And actually, the phase 2 trial for this was actually very successful. And they just came with the results 3 weeks ago. So this will invariably go to phase 3, hopefully the first FDA-approved drug for notalgia paresthetica. So, and we think, at least putting it all together, we think there are itch stimuli that might go through JAK or not, and we can activate. There are things that activate itch, but with difelikefalin, it mimics endogenous opioids that activate the kappa opioid pathway, and we think probably through mechanoreceptors actually sort of gates out itch. So in a way, it's a way to we scratch to suppress itch, and it's through the mechanoreceptor. So we think that this is a form of kind of pharmacological scratching, and hopefully results in less pathology within the skin. But it's interesting, even before all these recent results came out, I wrote this perspective for Neuron, because there were so many people asking me these questions. Why is itch suddenly big? Why does it matter? And why is pharma suddenly so big on it? So I wanted to kind of address it in a very succinct manner. And one of the reviewers actually commented, I'm not so sure it's translational or revolutionary, I'm sorry. But I said, I kind of pushed back. I said, it is pretty revolutionary in the sense that the hit rate has been so high with these trials and with such little investment from a funding standpoint, because this isn't like the most highly funded area of NIH at all. So the return on investment has actually been quite tremendous. And so it's a very, very exciting area, but it extends beyond that. If you actually look within the vagal ganglia, again, you see these itch neurons pop up as kind of top hits, even within the vagal ganglia. And we know that roughly 80% of the vagus nerve is actually sensory. Again, something that I never really thought. And so and then if Masato looks just using TRPV1 reporter mice, we find that you can't actually distinguish the vagal ganglia from the DRG. So they basically look kind of the same. So they seem to have a very kind of similar sensory machinery. But along these lines, the nalbuphine, the kappa opioid agonist and mixed mu opioid antagonist from another company actually had very good phase 2 data on chronic cough. And what we think is that cough is probably very similar to itch, and a lot of the kind of cellular and molecular machinery is similar. So we're starting to think of, are other sensations within the body much more akin to itch than we think? And so we know that we're studying somatic afferents in the skin, but then there are these vagal afferents. There are these spinal visceral afferents, which we know very little of what they do at all, and they probably innervate. So when we're looking at the DRG, we often are assuming we're reading this out, but we're forgetting about this part. So how does this all come together? This is I think is a very rich area of exploration. It's quite a frontier. I don't think people know much about this at all. If you go into the trigeminal ganglia, we find itch nerves in the trigeminal ganglia, which is not a surprise. But what's interesting is in this study, they looked in the context of migraine headaches. And as you go from mouse to human, they become much more peptidergic. So the human nerves, again, more polymodal, have CGRP, are very enriched for CGRP. Again, how are migraine headaches really kind of pro-inflammatory? Are they, how much is sensory and how much is pro-inflammatory? So I think this is a very interesting area, and we like to think of itch as itch is a big problem. We estimate anywhere from a $50 to $100 billion problem for pharmaceutical industries, but there are many, many disorders that have sensory pathology that are actually kind of ignored in the clinic. But I think what we can say is that itch is actually revealing how these are actually quite worthy conditions that we can actually potentially go out after. So, we started with asking what causes chronic itch. We then delved into kind of fundamental neuroimmunology. I think we've been able to inform and accelerate the therapeutic development and even design clinical trials towards FDA approval. But we've also been able to reverse translate to get at more other kind of fundamental biology. And, I just wanted to comment, here's the body and here's the brain, and there's a lot in between. But I think the key is understanding where are the frontiers. And I think there's been so much great work done within these specific domains. I mean, there's no question to me, neuroscience has been incredibly fruitful. But I think we want to understand, we were just focused from here to here, and it's just been a very, very rich kind of frontier for us. And, can we connect this all together? And that's kind of how I think, we can work all together through centers like this Brain Body Center. And, I just want to end by just thanking all the people who did the work. I highlighted the individuals who led the work that I showed here today, our collaborators, our funding agencies, and happy to answer any questions. Thank you.
Okay, great. So we'll take questions from the room first, and people online, if they have questions, just put them in your chat. I will take a quick look and field them as we go. So we'll start with questions in the room and then pivot to online.
Sure.
Hi, Brian. Amazing talk. My question is related to mental health and itching, because because it's bi-directional, right? The conditions like PTSD, there's a lot of itch sensation, at least from what I know.
Yeah.
But then you also mentioned that with some of these drugs, the distress aspect of it is reduced. So in your clinic or animal models, do you see this connection, and what level is it in regards to certain mental health conditions?
Absolutely. Yeah, unequivocally in patients, unequivocally. So I've actually thought that a lot of patients who presented to me, I had no doubt that they were itchy, but I thought they had deeply rooted kind of mood disorders because they were so profound. When we're able to actually get their itch better, they actually completely recovered. And so I have stories, I can tell you lots of stories. I had patients come in with lymphoma, that was undiagnosed for a year and a half, that were put on antipsychotics. So it's very, so they have a huge effect clinically. And that's something Scott and I actually talked about this. The brain is pretty scary to me. So I say, I can't do a lot of these behavioral assays to look at more mood defects or even cognitive defects, right? And we have to wonder how much of these peripheral stimuli actually can rewire even in the brain. People talk about even itch central sensitization. No one's proven that in the itch field yet. I know it's big in pain, but no one's proven that. It's just a concept we assume away from the pain field. So yeah, there's a lot that we're interested in. And furthermore, how does this then go the other way, right? So there's a lot of complex interplay. But yeah, I'm very interested in that, especially, with optogenetics and, stereotactic targeting of these areas.
I got it.
Yeah.
I'll give him this one.
That's okay, America.
Thanks, Brian. So in your talk, I think you nicely sort of alluded to this idea of itch being a cause or a symptom of something. So I understand, for example, that if you have an inflammatory condition, skin condition, that itches and you have that need to scratch, right? But sort of how do you think about sort of the advantages of having an itch where it doesn't really potentially lead to pustules or sort of a dermatologic problem? Like you're playing the piano and all of a sudden your nose itches and you just are fighting. You cannot stop playing the piano to scratch your nose, and it takes all sort of will to not to scratch your nose, because then you'll stop playing. So sort of how do you sort of think about these kinds of issues? Because I think during your talk, I probably scratched myself 15 times just listening to you speak, but I'm not going to get a dermatological condition about this. So what are sort of the advantages of this? Is it a grooming thing? Is it some residual? Just overall, how do you think about this?
Yeah, yeah, I mean, it's clearly protective against parasites, mites. And you see, if you have a dog, you'll see that dogs actually scratch quite a bit. And they actually, some of the biggest blockbuster drugs for itch are in canines. So two of them, IL-31 antagonist and a JAK inhibitor. So yeah, it is protected. And there is actually a contagious component that's protective. So socially, if I am scratching, it's presumed by you instinctively that I actually might have scabies or body louse or something, and you start scratching so that you don't get infested, I think. So I think there is a contagious element to it that is socially protective. The other interesting thing, though, is it's very socially aversive. And Scott and I were talking about this. Like the mice don't like to be around other itchy mice, like humans. If someone's scratching in the subway, people don't tend to sit next to them. So there's an incredible amount of actually social stigma, but it's very hardwired into us. So I think it is protective, but it's protective against things that we already kind of know we don't want to be around. So yeah, but the other thing about your piano analogy is that there's a lot of suppression. So whereas a concentration, cortical suppression, there's lots of ways that itch actually can be suppressed throughout the day. That's what's very hard to study in patients. So interesting, we actually done studies with MIT where we actually use touchless technology to count scratching in humans, but we also have video cameras. Patients are consented. They actually, it truly is a reflex. They scratch in their sleep. They don't know it. And there's some discrepancy in terms of what they think they're doing and what they're really doing. So yeah, there's a lot of, it really kind of gets at also kind of fundamental questions of what does it mean to be conscious and unconscious with regard to what is a reflex but doesn't have to be a reflex, can be initiated by a thought or someone talking about it. So there's a lot to be still studied, I think, in it. I think it's a pretty fundamental, it brings out some good fundamental questions.
So I'm already thinking about a million things that we can do together. This is a great discussion. I'm going to pivot to some of the online questions. The first one is from Tuhu, who asks whether there are biomarkers, perhaps molecular biomarkers that could be used to characterize itch.
That's the holy grail. Not every pharmaceutical company wants to know. It'd be great. It's hard though.
So you're talking to a psychiatry researcher, so you have none.
Exactly.
The second question is from Olivier Massot. Where does the lymphatic system fit in this paradigm of itch?
Yeah, I don't know. There's lots of things that people ask about. We don't know with the lymphatic system. We don't know a lot about the vasoregulation in general, too. The blood supply and how that plays into it. And we also don't know how much the autonomic nervous system does in this. And we know that there's a lot of autonomic triggers probably, but we still don't even know how they, I only talked about somatosensory. And then I'm just going to say a bunch of stuff we don't know. Microbiome, we don't know either. So there's a lot of hot topics in itch that have not been explored. So, yeah.
Okay, I'm going to get one more in here. Actually, we've got three more. Next one is from Supinda, who wants to know, how do you currently balance anti-inflammatory and anti-neuropeptide meds for AD treatment in children?
Yeah, it should at this point be really mostly the anti-inflammatory agents that, in my opinion, which they all do, have some neural activity. So in children, dupilumab is approved now for down to 6 months. So I think that's a game changer. In terms of atopic march, there's some suggestions from preclinical studies that you might even prevent the onset of asthma and other subsequent allergic disorders. So for kids, that's going to be the strategy. So far, neurologic strategies we have yet to be determined. And as I showed the kappa opioid, difelikefalin is kind of the first one.
We have 2 more questions, if that's okay with you.
Absolutely, yeah.
Hi, Brian. Thank you for a great talk. Can you elaborate on side effects of systematically targeting JAK in patients?
JAK inhibitors, yeah. The issue with JAK inhibitors systemically, we know this, is that there's a significant, but small, but very significant increase across the board in a lot of things. Cardiovascular events, blood clots, cancer, infection. So they are immune suppressive. That's the issue with them. The question is for us, could you just target the nervous system? There are potentially ways you could do that. But they weren't conceived to do that. They were really tailored for autoimmunity. So that people were trying to get the anti-inflammatory immune suppressive properties from them. But yeah, so that's an issue with the systemic JAK inhibitors. And they all have a FDA black box label because of that.
Thank you. Last question then. This is from Crystal. For the dupilumab, I can't say the name of that
Yeah.
trial, have they looked into if itching resume or increase back to normal baseline after stopping using drug?
Yeah, in general, I have a lot of experience. In atopic dermatitis, it comes right back. So there's no remittive property. Yet to be determined for these really more neuropathic conditions. Because the kind of question is, these people weren't itchy, and then they became itchy, and it's quite persistently itchy, months, years. So is there any way you can turn it back? All right, is there any way that you can rewire them? Unclear as yet.
All right. I guess
Great.
if there's no more questions, then we'll wrap it up.
Great. Thank you.
Thank you.
Thanks, everyone.